Good evening, everyone. I am delighted to welcome you all to the today insightful online session. Where last session we have to discuss about the ethical consideration in herbal observation study. In today's session, we identifying endpoint safety, efficacy, and quality of the life and real world trials. I miss Sakshi Dabe and I will be your host for the today evening. We are truly honored to have Dr. Sriharan sir with us. I distinguished expert and researcher. They will be a guide us through this session. Now I request Dr. Sriharan sir to the carry this session forwards.
Hi all. Yesterday class we see about ethical consideration, how in herbal medicine take place, how we get the benefits sharing. Likewise, we see about ethical related in yesterday class. So today class is going to see about safety, efficacy and quality of life in our real world trial or otherwise called as observational study. So we are going to look on it, but on the basis of endpoint we are going to see on it. So foundation of clinical research. So, why the endpoint is matter? So, endpoint is one of the important core element in a clinical trial because why means it will measure the outcome. What outcome we are going to measure? It will tell about that. And endpoint also not only the endpoint will not stay only in the laboratory condition. It also reflect in clinical setting also. So your endpoint not should be in a lab animal data or lab in vitro or in vitro stage. So it want to expand to the clinical setting also. So that is why important the endpoint is. So selection of endpoint why important means it will give the shape to the our sample size. So the endpoint give the shape to our sample size, and also it will be useful for the risk benefit and regulatory decisions also endpoint play major role. So the three pillars of clinical endpoint is first one is safety, efficacy, and quality of life. So this is the main pillars of the clinical endpoint. So here the safety endpoint will detect the adverse event, serious complication like toxicity, like will tell about based on that. So efficacy endpoint measure the therapeutic benefit, what actual clinical effectiveness, it will tell about that. So quality of life endpoint is patient well-being. So how the patient daily performance, likewise, it will evaluate that. So this is the three pillar of the clinical endpoint. So understanding the endpoint hierarchy. So you know about that the endpoint should be primary, secondary, the objective should be primary, secondary. We will see a lot about a previous class itself. So here the primary endpoint you know about that it is a proof of concept. You in single word you want to know means it is a proof of concept. It will answer the main research question of our research. And secondary endpoint is the additional supportive. So it is add-on. So supportive evidence to give to your research endpoint. That is the secondary endpoint. So exploratory endpoint is generating for future research. It will generate the hypothesis. So this is a primary versus secondary endpoint. So today we are going to see about chapter wise chapter. So today the first chapter is safety endpoint. So before we are proving efficacy, safety is first. Why means we want to prove the safety. Then alone we will go effectively in a clinical setting to test in the human being. So before proving efficacy, the safety is a must thing. So it will take place in a rigorous safety monitoring. So the safety monitoring is not in a single step. So in a single word saying it is not a one day process. It will take large time. So alone it will help to identify any harm, safety signal, any deviation from the our observational drug or anything, it will tell us. And based upon this safety monitoring, we also prevent the harm and maintaining the patient in a good condition in a safety manner. So safety monitoring is very important. It will take place in all the steps during a baseline character before starting the trial and after screening one, treatment during treatment, screening two, screening three, and follow up is a post treatment. The safety monitoring is a long process. In single way, you want to say, you want to know means it is not a single step. It is also not a one step. Once in a once is not. So it will come last up to the last end of the study, and after the post of the study also it will continue. So category, you know about the safety category. So in safety, you may know about the ADR, what is ADR and how it should be reported. Likewise, in clinical trials also there should be adverse event, serious adverse event of the special interest. So these are the events are very important thing while doing in a research in a drug or herbal drug. Why I'm saying means we want to give the report to the regulatory authority. So the safety monitoring is main thing. So the adverse event, it should be a mild, moderate, or light mild. So likewise, the adverse event takes place. It also adverse event also classified as major adverse event, minor adverse event. So secondary is serious adverse event. So serious adverse event, we want to concern focus mainly on this alone. Why I am saying means the serious adverse event are hospitalization, lead to life threatening or sometimes death also. So this is a serious adverse event. So adverse event of special interest. So adverse event of special interest is nothing but while doing it is a predefined event. It will cause if you done research on this drug means we have a history of that, we have a mechanism of that, it will cause definitely. So that is it not be like a random cause, random event like adverse event or serious adverse event or random event. It will cause as a randomly. So it is not a random event. Adverse event of special interest is not a random event. It is a predefined event. We know well known about that. So that is a event of adverse event of special interest. So other thing I want to also tell that while doing in a clinical research or clinical trial, we want to report this to the regulatory authority is a very important. So there should be a timeline on that. So 7 days, 15 days, 24 days. So if you have a serious adverse event, you want to immediately report to the regulatory authority within a 24 hours. That is a 24 hours as a time period. So adverse event also have a major, minor. So major should be reported to 7 days. Minor should be reported to 15 days. So this is the timeline. So this is a safety assessment tool. You know about that casualty assessment in ADR. It will play a major role. So safety assessment is very important. It will be classified by 5 stages: mild symptom, moderate symptom, severe symptom, life threatening. So death related to adverse event. These are grading. You are giving grading. What are the adverse event or a serious adverse events are coming to your trail? You are grading that. So MEDRA coding. So MEDRA coding is one of the important thing in pharmacovigilance. If you go to any, if your future jobs should be based upon pharmacovigilance means, so MEDRA coding is main thing. You want to more concentrate on that. So why means it is a hierarchical branch, preferred term. Likewise, it will have a coding center and preferred. Basically, in MEDRA coding, it will the first branch as a preferred term. So preferred in the preferred term, it will tell the respiratory system, cardiovascular system. Likewise, it will go under subclass. It will have. So this is the MEDRA coding. Solon we called as a hierarchy branching. It will be in a subgroup in each section. Likewise, it will call. So MEDRA coding is one of the important thing coding the adverse event. So laboratory based biomarker. So why we are using laboratory based biomarker means it will show how the body organs are responding to our treatment. So this is very important. So based upon this laboratory based safety biomarker, we get the yearly warning sign of any toxicity. We want to do any dose modification. Likewise, we can get yearly sign. So our laboratory based safety biomarker so mostly focus on main organ only. It monitor main organ. So main organs means what? It will monitor the blood. So blood is main thing. Second thing is liver. Third thing is kidney. And last thing is heart. So this is the main organ. So in blood, we will monitor the CBC, HbA1c, HB. Likewise, we will monitor. So in liver, we monitoring liver, kidney, we will monitor ALT, AST. I got the liver function test, kidney function test. In cardiac, we will monitor prothrombin time, INR. Likewise, we will monitor. So why we are monitoring means, so monitoring this, how the our drug or product, it will in our organ health and drug safety. So likewise, we are monitoring on that. So this is one of the thing. So time frequency of assessment. So there should be a time. Why means before itself I'm say safety is not checked only once. It must be monitored thoroughly throughout our study and after the study also. During study phase 1, phase 2, phase 3. After the study, that is a post marketing surveillance, that is the phase 4. So baseline, before study starting we are evaluating. So during how the before patient condition. So during treatment, any toxicity, any profile of the drug pharmacokinetic, pharmacodynamic, we are evaluating. End of the treatment, we are identifying any renal impairment patient, how they are taking, how their toxicity may build up, and how we are, what are the requirement for renal requirement and hepatic requirement. Likewise, we will see about the end of the study. So follow-up period is a post-marketing surveillance, post-study. So some herbal drug have a delayed side effect. So it will not reflect in our phase 1, phase 2. It will after the study, during follow-up period, it will appear. So delay side effect or relapse. Something happened, relapse, it will come reoccurrence. So likewise thing. So this is a time frequency and safety concern. So safety monitoring in a special population. So the special population is very different from the normal individual adult. Why means their body react differently to the treatment. So there are special population, you know about that, pediatric, elder, pregnant. So in pediatric, if we take means their organs are mostly immature alone. So we want to concern that. Elder person, we will have a polypharmacy, they have a organ failure, sometimes they have a low observation, low absorption, likewise will have. So another, the last thing is pregnant lady. So pregnant woman means we want to monitor, dual monitor. We want to monitor the pregnant woman also and her children also. So this is a safety monitoring in the special population. So key safety metric. So key safety metric is what means it giving a data about how your treatment done during your trial. So it giving a data. So likewise treatment emergency, emergent adverse event, serious adverse event, treatment discontinues, dose modification. So treatment emergent means it will arise after the treatment. How many patients get experience this? It will observe that. So this is the observation value of treatment emergent adverse event. So serious adverse event, you know about that. Hospitalization, death, life threatening. Likewise, it will happen. Treatment discontinuation. So due to major side effect or any some issue related to the drug, they will discontinue the treatment. So the treatment discontinue also we can learn about tolerability data. So based upon the treatment discontinuous, we can easily identify and validate the tolerability. So dose modification. So dose modification should be reduced dose, skip dose. Likewise, we want to monitor based upon their condition. So here the dose modification also very useful for clinical manageable. So sometimes which should be optimal dose, which should be initially start. Likewise, we can identify based upon this safety matrix. So second chapter is efficacy. So efficacy means how the treatment achieved therapeutically benefit. So that is the efficacy. Achievement of therapeutic benefit is the another name of efficacy. So the efficacy clear core elements are your efficacy should be clinically meaningful and scientifically sound, and your data should be reproducible. So that is main important thing. And you should be, your efficacy should be a measurable one, an achievable one. So not should be unmeasurable or unachievable one. So these are the core element in clinical efficacy. So in clinical normal RCT, the efficacy are mostly predefined endpoint alone. So the type of efficacy endpoint. So time to event, it's time to event, you name itself, you know how long the time to event happen. So that is endpoint. It will tell how long it takes. Likewise, it will tell before the our definite outcome come. So this is time to event. So binary outcome is different from that. It will tell the whether our outcome is success or failure. In simple word, we want to tell means it is a yes or no question, yes or no outcome. So whether the patient achieved or not, so that is the binary outcome. So next to the binary outcome, the continuous measurement. Continuous measurement mostly in a numerical value. In continuous interval, you will get the idea about that numerical value. So numerical value means what? Blood pressure, CBC, cholesterol. So this is the continuous measurement of the patient. So based upon this continuous measurement, we can get the how the degree level of benefit to the patient. Likewise, we can get. So composite endpoint is different from apart three. Why I am saying means it will group the three multiple outcome in a one group. For example, what I'm saying means in simple language I'm saying composite endpoint. You cardiovascular disease you take. So in cardiovascular disease you are grouping heart attack, stroke, and cardiovascular death. So three is the three thing is a multiple outcome. So you are grouping into single measurement. So based upon that, we can get the full picture of cardiovascular event. Overall benefit also we can identify using this composite endpoint. So so another thing is overall survival. So overall survival is one of the golden standard technique which should be followed in a life-threatening disease like cancer. They mostly do. So this overall survival, basically how it should be measured means the time from randomization until the death or any event cause. So that is, it is a time between randomization to death until the death or event cause. It will measure that. So here you know about that. Overall survival have no intervention need. So we no need no intervention. It will say whether the patient survey or not. You will survey or not. It will give the data about it. So based upon this we can improve the lifespan of the rare disease like cancer. We can improve on that. So it will also give the proof for our treatment is benefit. This is the survival data. So likewise it will give the proof of benefit. So overall survival is basically is not be a single day process or single month process. So it will take long time. So overall survival is a long time follow up we want to need. So in the and one more thing I want to add means overall survival is also universally accepted one of the method. So it is one of the value method also. So it's used in a endpoint section. So another thing is progression free survival oncology. So compare followed by the previous slide, it is also one of the survival method, survival endpoint. Here, what is different means they measure time from the randomization until the death. But here we are measuring what means the time from the randomization to either the disease how progressive, either how the decrease is progressive or the death. Which come first? We are going to see about which come first. There we see how the timeline between randomization until the death. Here we are seeing the randomization time from randomization either the diseases come first or death is come first. We are seeing that. So it is compared to overall survival. It is not much more long follow-up is need. So 1 month is enough for that. So more widely they are mostly used for progressing free survival in oncology alone. So it will give the drug effectiveness, read out the drug effectiveness very early, often used for accelerative approval. We see about in upcoming slide what is accelerative approval. So it often taken a 1 month is enough. So basically the pharma company used this progressing free survival for the getting faster approval from the regulatory. But one thing is have a challenges on it. What means assessment. So this assessment is very some difficulty faced. Why means the assessment is otherwise called as a receipt. A receipt is nothing but the full form I'm saying response evaluation criteria in a solid tumor. So this is one of the method for measuring this progressing free survival. So here if you are measuring the patient any cancer patient means your imaging or your biomarker showing there is no growth on the patient means the patient considered as a progressive free. So for another example, for the patient have any tumor growth or new metastasis means they consider as a progressive. So based upon that, we can get a easy approval. We can easily identify the drug effectiveness very early. So it also tell about the clinical relevance. So clinical relevance is what means if the patient have stable symptom, the patient have a stable symptom, they have they continue with the normal life, but the tumor growth is increasing. So it differ from they not affect it will not affect the symptom, but the tumor growth is decreasing. So it also tells the clinical irrelevance also. So response rate endpoint. So response rate endpoint itself itself you know about that it will measure the improvement in disease after our treatment. After our treatment, how our response is increasing. So objective response rate is whether the patient achieve partially or completely response based upon our image or laboratory, we are calculating that. So another one is disease control rate. So disease control rate is give the percentage of how completely respond, how partially respond, how the stable the disease. It give the percentage to that. So through this disease control rate, we can see about how the disease will progress, how the disease is not progress. We can see that. So complete response rate. So complete response rate, you know about that. Completely disappear of disease. So that is a complete response rate. So, surrogate endpoint in clinical trial. So, surrogate is nothing but it will measure the direct clinical outcome. It will measure the direct clinical outcome instead of waiting for more than year or more than month. More than several years. It will say whether it will improve or not. So, it will say in a early manner. So it should be in a biological, have a four-step biological plausibility, whether the biological link to the disease progressing, it will tell, and statistical association with the real clinical benefit and treatment effect capture. How or treatment effect it will have capture. It will tell, and it also accepted by regulatory validation also. But all the surrogate endpoint is not reflected in clinical setting. So we want to consider that also. So cardiovascular efficacy endpoint. So for example, cardiovascular is considered as a chronic disease alone. So it will slowly evolve. So long-term observation is need for the cardiovascular seat. But using this surrogate endpoint, we can get faster assessment. We can get faster assessment. So why I'm saying means first previous itself I'm saying again also saying. So all the surrogates are not important or not valid also. Why means we use some surrogate biomarker which should be effective in our trial, but it will not reflect in our clinical outcome. So, surrogate have also some limitation and challenges also. So here the cardiovascular efficacy. So previously we see about the composite endpoint. So here we are using as a composite endpoint. So grouping a cardiovascular event like stroke, cardiovascular death, and cardiovascular event. Likewise, we are grouping multiple outcome in a single event. So based upon that, we will get the overall picture of the clinical benefit. We will get overall how it will happen, how will be, what is the proxy. We will get the overall picture. So plus the overall picture and also secondary, it will tell about hospitalization of the patient for the heart failure, any stunt, any bypass surgery has been done. Is also secondarily tell about that. So based upon this cardiovascular efficacy endpoint, we can track both fatal and non-fatal stages in the disease. So one of the major thing is we can understand how the our treatment prospect in real world outcome also we can access. So functional outcome measures. So functional outcome measure is nothing but it will tell about how the patient feel and function in a daily life. So it will measure like a 6 minute walking test is based upon their exercise capacity, forced expiratory volume. Patient like COPD, they have a forced expiratory volume. So the airflow have limitation. Likewise, we can measure. Disease specific classes, scale is basically for specific disease like Parkinson, they have a motor inability, so it can validate and identify that. So this is a functional outcome measure. So biomarker endpoint, the role of a biological biomarker. So the biological biomarker it will measure and evaluate the disease status and how the treatment response. It will tell the disease status and the treatment response. So how the drug, how the herbal product working at the our molecular pathway, lipid profile, protein binding, how it will work, it will tell about that. For example, I'm saying HbA1c, you take that HbA1c. So it is act as one of the biomarker. For what? For controlling blood sugar control. And another thing is LDL. So LDL cholesterol here act as a predictor biomarker. So it will tell the risk of heart attack or stroke, it will tell. So this is one of the role of the biomarker. So all the biomarker not accepted by regulatory authority. Why I'm saying means we need a strong evidence to accept our claim to the as a biomarker to the regulatory authority. So strong evidence to be need. So all the biomarkers should not be valid and should be not considered. So that you want to also consider that. So statistical consideration, it is one of the part in efficacy. So sample size, you may know about the power of sample size. It's 80 percentage or 90 percentage we want to set. So sample size determination we see about lot on our session. So here the multiplicity control. So here multiplicity control is what nothing but so we are testing a multiple endpoint. We want to think that we are testing multiple endpoint. So we are also testing on subgroup also. So sometimes we want to need adjustment. So adjustment we want to need to adjust the population. So adjust a number of sample want to need due to the confounding or anything. So if we not adjust mean that should be a type 1 error risk. Type 1 error risk is a false positive. So false positive will be increased. Without adjustment we can do means false positivity increase. So this multiplicity control give the study validity and integrity to our efficacy. So the multiplicity control is one of the major thing. So here a non-inferiority margin. So non-inferiority margin is a comparison margin. So what I'm saying means it will compare the new treatment against the existing one. So my drug, likewise X drug is effective than the existing drug Y. Likewise, we will give the comparity on that, and it also say whether the new therapy is good enough to use or not. Likewise, it will tell, and it will tell the what is the difference between new therapy and old therapy. So it also tell about that. So in multiplicity control, I missed the point on a hierarchical setting. So hierarchical, it should be a predefined setting. So Benophoria is nothing but it divided by alpha. Likewise, it will be statistically conserved. And gatekeeper method is logically we are grouping. So these are the methods are used in multiplicity control. So quality of life in endpoint. So the quality of life itself, you want to know, you may know about the PRO, patient report outcome. So that is a patient voice in the clinical research. So it also play major role on that. So the component of health related quality of lives are physical functioning. We can using their mobility, self care ability. We can also take a PRO on that. And the emotional health, whether the physiological well-being, their mood swing for psychiatric patient, we are content. So social well-being, how they are mingled with the environment. Likewise, the role limitation, how their capacity, how their our mobility. Likewise, it will tell. So symptom burdens based upon the disease. So this is the component of health-related quality of life. So quality of life instrument there should be a certain form. We have seen much morely about in our previous session itself. So it is a SF36 is one of the short health form.
used for the physical functioning role of body pain. Likewise, it is one of the form. So EQ5 is a EQ5 dimension. So it will tell about mobility, self-care, useful activity, pain discomfort of the patient in a quality life instrument. So it is one of the forms you know about that. So here the disease specific quality of life means the PRO should be based upon specific disease like asthma, like cancer, like psychiatric illness. Likewise, we have a individual disease, individual form for the individual disease, individual PR for the disease. So that is a disease specific. So patient report outcome measure. So you know about that. So the patient voice also very important. Why means it will tell the health status without the clinical interpretation or clinician. So the PROs may play major role in decision making also, regulatory decision making also. FDA what initiate one of the scheme we will see in upcoming thing. So PRO play major role measuring outcome. So using that we can measure the symptom severity scale, likewise how the pain reduction and fatigue improvement, sleeping quality. Likewise, we will measure that. So functional assessment status, how they are doing the basic activity of day-to-day life, how the instrumental activity like shopping, cooking, praying, going to temple. Likewise, their mobility. Advanced activity like hobby, engagement in public society. Likewise, so here we are going to the previous slides are we are see before, but this is different from that. So we are minimally important difference. So minimally important difference is nothing but any smallest changes in our treatment outcome. Patient could identify them as a personally meaningful outcome. They itself consider as it is a personally meaningful. So that is a minimally important difference. So based upon this code, doctor can also change their care plan also. So this minimally important difference play major role. So they have a certain method. So anchor based method is nothing but it will evaluate the what current versus old condition. For example, it is a globally accepted one. For example, what I'm saying means, if you ask the patient means, did you feel better? Likewise, you are asking. Patient saying, little better. So likewise, your current and old condition, that is anger based method. So descriptive method is based upon the standard deviation, the standard error. Likewise, it should be based upon a statistical manner, the distribution method. And another thing is patient preference. So patient preference is nothing but is patient voice, how they are meaningfully in they treat this outcome as a very important. Likewise, they are saying. So the smallest change is very meaningful in their point of view. So that is minimal important difference. So here, what we want to know that what we want to know means, so all the number and p values are here not important. Only here what is important means yes. Likewise, if you feel better, yes, I feel better. If you have any changes notice, yes, I changes notice. So based upon this questionnaire answer alone, not should be based upon how many numbers and numbers p values are not play major role on that. So that is a minimal importance difference. So timing of quality of life assessment, you know about that baseline, yearly, mid, end, follow. So there should be a screening before screening should be baseline, yearly, midterm, and treatment. After the follow, it will be a long process. So fourth chapter is real world evidence consideration. So in RCT or control trial, it will tell whether the treatment is work in a control environment or artificial environment. Can it work? That is their point of view question. But in real world evidence, it will tell whether it actually work on our everyday life or routine clinical use. It will give the data of that. So that is the clinical trial, from the clinical trial to the real world practice. So it will tell how our routine clinical life and day to day life data are very useful in our research. Likewise, it will tell about that. So real world data source. So in observational, we want to get the data from that. So the source of data, we know about that. We will get the electronic health record data, claim database, disease registry, and digital health data. This is a source of data where we are getting data for our study. So endpoint selection in a real world study. So this is very important thing you want to consider. Why means in control environment, the endpoint I am previously say it is a predefined one. But in real world study, it depend upon what data already in our database source. So likewise, this is already the database or electronic health claim database, disease registry, digital health data. So this is the already in database. So it will capture the our it will capture from our healthcare database alone. So our selection must be, it should be in a three manner. You want to understand that. First thing is it should be a clinically relevance. Second thing is data should be practicability. It means our data should be futurely it should be in a clinical setting integration only. So your data should be in a practicability. And another thing is meaningful health outcome. So achievable, meaningful or benefit health outcome you want to deliver. So that is the endpoint selection in a real world study. So the challenges. So the what are the major challenges mean? You know about that. So maintaining accuracy and consistency is a very challenges. Why means we get the data from the multiple resources like electronic registry, claim database. So the data come from multiple registry. So data completeness, incompleteness also one of the cause for the challenges. Why means inconsistent entry, missing data, which lead to the data completeness. So measurement standardization. So measurement standardization is nothing but the every hospital or individual hospital follow the individual rules, individual treatment style, individual standard operating procedure. So based upon that variation, the measurement standardization takes place. So confounding by indication. So confounding by indication means, for example, the patient we are choosing a patient for the observational study. We are getting the patient will get a new drug for new disease. Likewise, it will happen. So it will cause a confounding. So it will be solution by the propensity score matching. So outcome validation means the outcome validation majorly come for the cardiovascular system alone. Why means in cardiovascular system there should be a stroke, cardiovascular death, bypass, stent, stenosis. Likewise, it will have a multiple classifications. So there should be classification should be takes place. So we want to verify using coding algorithm. You can verify. For example, the patient have stroke means you want to cross check with the imaging report, MRI, CT. Likewise, you want to cross checking. So that is outcome validation. So pragmatic trial design. So pragmatic trial design is nothing but whether the drug or any herbal product that proved well in a controlled environment. So control, but in our real world scenery, how it going. Likewise, we are accessing their effectiveness in a normal routine care. We are accessing their effectiveness in a normal routine care. So it will fill the gap between our traditional RCT and observational studies. So this is one of the initiative on that. So it will take place without control environment, without restriction. Like in our city, it will tell in a normal clinical setting. But the main goal of our researcher is how the treatment work in real world. That is the main goal for our pragmatic trial design. So they have a one of the domain that is called as a precis. So precis is nothing but pragmatic explanatory continue indicator summary. So it will based upon this pragmatic trial design. It is one of the assessment. Precis 2 is one of the assessment. So this assessment will tell about how we want to include the broad inclusion criteria, how will be create, and flexible treatment delivery, flexible visiting, flexible scheduling, how the routine data collection takes place, how the we are clustering our randomization, subgrouping, like is how the outcome. Here the outcome mostly focus on how the patient responds, likewise quality of life, PRO. The outcome is mostly is the background of PRO. So that is a pragmatic trial design. So healthcare utilization endpoint. So it will measure how the patient used the healthcare service, like hospitalization rate, length of stay, emergency visit, and cost saving. How the patient are using healthcare due to this treatment effect, how they are using. So these endpoints are majorly used for economic and economic factor or any operational setting factor. We can use that. So chapter 5. So chapter 5 is a regulatory aspect. So you know about that regulatory like FDA, EMA. It will evaluate the clinical endpoint based upon how well they are demonstrated in a real clinical setting. So the endpoint is very important. So our endpoint also truly it will be reflect on clinical setting. So before itself, I'm saying the surrogate biomarker or any biomarker, not all the biomarkers should be validated by the regulatory authority, which should be reflect in a clinical setting and clinical integration. So that is the main important thing we want to consider. So then alone they will accept. So not they if you want to accept your biomarker, you also give the strong evidence on that. So that is also one of the important thing. So FDA guidance of clinical endpoint, you know about that. Sorry, FDA guidance of clinical endpoint. So first thing is it should be a clinically benefit and it should be measured property, like it have a validated reliability. Likewise, it will have measured property. So here you know about clinical benefit and measured property. So here the interpretability. So it is different. You may not know about that. So here interpretability is nothing but your result must be a clinical significance, not only just like in a statistical significance. It should be main first thing is a clinical significance is very important. So that is an interpretability. So data quality, if you get data quality is the data come from the multiple sources. So you want to verify the every resources and accurately monitor that. So based upon that, we can minimize the missing data, incompleteness. Likewise, we can minimize that. So accelerator approval pathway. So accelerator approval pathway in previous slide we see about the overall survival and another one is progress-free survival. So we see on that. So here that any life-threatening disease like cancer, likewise any disease, the surrogate endpoint can be accepted and it will give us the accelerator approval pathway. So here the using a progressive free survival assessment. Based upon their assessment, they give a accelerator approval to your drug. But one thing you want to confirm that is a confirmation requirement. So the sponsor, researcher, or investigator want to prove whether their drug also safe in the post marketing also. If the drug is safe, it will be continue in the market. But it will be have any failure in post marketing means it lead to the withdraw from the market. So that is the accelerator approval pathway. So it is very useful for the pharma company. So here the patient-focused drug development. How the PRO play major role. Patient report outcome play major role. It is initiated by FDA. How the patient perspective directly, which will directly reflect in regulatory decision making. So used upon this patient-focused drug development based upon the collected data from the PRO, how the patient voice. Likewise, it will tell about that. So EMA. So after FDA, EMA. So EMA, how they select the endpoint, how they give the justification to endpoint means your endpoint should be a scientifically sound. And another thing is it should be have a validated evidence. There should be a no invalid surrogate marker like this. It should be validated and statistical analysis plan. So likewise, a normal clinical trial requirement for endpoint followed by the EMA. So labeling and claiming. So apart from this, labeling and claiming is nothing but how your endpoint depends on certain strongest evidence on that. What means your endpoint have a greater evidence to claim the labeling on the evidence like that. For example, I'm saying means type of claim. So type of claim means superiority claim. So superiority claim is nothing but it demonstrates how our outcomes so better from control versus sorry. So how our outcome is improved was control versus study environment. It will tell about that. That is a superiority claim. So non-inferiority claim is a comparable evidence. So like comparable evidence means what means it will tell the benefit of drug from new drug to existing drug, old drug. It will give the comparable evidence on that. So QOL claim is based upon the patient responsiveness we are getting, how they validate PRO data likewise. So subgroup claim is based upon the subgroup classification, how they are pre-specified population, how they are considered the effect in the treatment. Likewise, it is a type of claim in labeling and claim. So come to the sixth chapter is a practical implementation. So, building a strongest endpoint, there should be some strategy. So, the followed by the first one is define objective. Your objective should be in meaningful and clear and detailed. And after that, selected endpoint. So, selection of endpoint should be a clinically relevant, and the data should be practically available and practically possible. And last thing is it should be have a meaningful health outcome. So that is a selection of endpoint. So develop the protocol based on that. So train the concern faculty, training, staff to the endpoint evaluation and monitor the quality of data, track the quality of data to avoid the missing data, to avoid the incomplete data. And after that, analyze the result based upon the statistical analysis plan. So this is the building strategy of the endpoint. So ensuring the data quality and completeness. Here, why I'm saying means in RCT they have a pre-specified, clearly measurable. They have, but in observational study they are totally different. So it will get the data from the day-to-day life. So the data monitoring and quality is very important. So here, assessment schedule means put a flexible schedule or flexible visit to the patient. So due to that, we can prevent the mystery endpoint assessment. So assessment scheduling is very important. Based upon flexible visit of the concerned patient or concerned participant, we can prevent the mystery endpoint assessment. So electronic capture. So validate electronic capture system. Adapting the validate electronic capture system, it will capture the data collection and it will self-evaluate. If anything incomplete means it will reflect to our mail or reflect to our mandatory area. So that is electronic capture. Central monitoring is a dashboard. So what dashboard means? It will tell whether the risk or benefit issue arise from our trial. It will reflect on our dashboard. So you give the first priority to that to rectify that, to control that. So that is a central monitoring trial. It will tell. Central monitoring is nothing but it will tell whole overall process of our trial. It will tell about that. So training and standardization. So giving a training to the concerned person, staff, likewise, initially before starting activation of the trial, giving initial trainings in and conducting refreshing training, likewise, real case scenery and performance feedback. Likewise, it will improve the training for the individual. It will be effectively in data collection to accelerate our trial. So managing missing data. So managing missing data should be like assessment of flexible schedule. Likewise, how the missing should be based upon mostly here should be a patient report due to the due to the scheduling alone. So use remote virtual consultation. So we can avoid the missing data on that. So today takeaway means how the endpoint, how your endpoint should be suited for our trial objective. You want to align that based upon the strategy, and uh, and put the endpoint which should be feasibility, should be data should be practically available and validity. So the PRO, patient voice, is how the quality of life data play important role in regulatory assessment. Likewise, you will see about that. And data quality and documentation, use assessment scheduling, electronic capture, likewise the central monitoring, you can achieve that. So the conclusion of the our today's study is, so any successful trial, the safety, efficacy, and QOLO is very important. So based upon that alone, we can move on our study to the in a large manner. So it also be a reproducible, and it also should be available in a clinic setting or a clinical setting also. So the safety and efficacy, quality of life assessment is very important while doing any research and observational or normal state. It's very important. So thank you for your patience.
Thank you, sir, for the sharing their valuable, insightful on the identifying endpoint safety, efficacy, and quality of the life real world trials. Your expertise has been truly in this session. Sir, some question asked the audience. First question is, what is the impact of the real world evidence on assessing long term safety
Accessing long
outcomes
perspective.
chronic
Yeah.
disease treatment?
Mm. Why means here they asking for the long-term safety outcome. So chronic disease. So here what we see about in long-term disease treatment category means we are seeing composite endpoint. So accessing the real-world evidence in long-term means the composite endpoint play major role. For example, the chronic disease treatment means you know about the cardiovascular disease. So cardiovascular disease in cardiovascular system there should be a multiple disease, stroke, heart failure, or cardiac death, or stenosis. Likewise, it will happen. So individually, if you see the individual outcome means it will get a long time and it will take a long manner. So we are grouping these kind of the chronic disease. We are grouping these all the diseases into one single group. So multiple outcome to the single group. So based upon that, we can effectively identify the data. We effectively collect the data from the our research observational study in a real clinical world setting. We can effectively collect the data. So the composite endpoint play a major role, and it also give impact in our chronic disease treatment plan or treatment observational study. Based upon the chronic disease treatment research, it will very useful. So composite endpoint play major role on that.
Okay, sir. Next question is the how is the data privacy maintaining while the collecting a sensitive safety information?
Yeah. So here in observational, if you ask observational study means yesterday itself we see about how the sensitive safety information should be collected, how should be data. So there should be a explicit agreement form. So before that, you will say about what will be shared and what will not be shared. So it should be attached in our informed consent form. So based upon that, our data should be protected, what should be published and what should be will not publish. Our author want to stick to their explicit agreement. So based upon that, we are protecting the data privacy and maintaining the data privacy through that explicit agreement.
Okay, sir. Thank you so much.
Thank you, ma'am. Thank you for hosting.